and that will lead to no functional tumor suppressor effect, and that will lead to malignancy. The classic example of a tumor that illustrates the two-hit origin is retinoblastoma. This is the tumor in which the two-hit origin of cancer was originally described. It is a rare childhood eye malignancy, and 40% of cases are hereditary. In these cases, children inherit one mutated gene that's found in all cells of their body at birth. This is an example of a germline mutation. They then at a later point in childhood get a second somatic hit that takes out the second copy of the tumor suppressor gene. Because they are born with one abnormal form of the tumor suppressor gene, cancer requires only one somatic mutation in order to develop. Therefore, these children develop frequent and multiple tumors, and they develop tumors at a younger age. Contrast that with the sporadic form of retinoblastoma, which is non-familial. Children who develop this form of retinoblastoma need to have two somatic hits that take out both copies of their tumor suppressor genes, and two mutations in the same cell is a rare occurrence. Therefore, these children do not develop multiple tumors, they develop a single tumor, and in addition, the tumors usually occur at a later age. Another example of the two-hit origin of cancer is HNPCC or Lynch syndrome. HNPCC stands for Hereditary Non-Polyposis Colorectal Cancer Syndrome, and I discussed this in detail in the GI section, but for our purposes here, you should just know that this is an inherited colorectal cancer syndrome that involves a germline mutation in DNA mismatch repair genes, and these mismatch repair genes are another example of a tumor suppressor gene. So in this disorder, you have a parent who passes on two copies of a tumor suppressor gene to a child. One of those copies is abnormal because of a germline mutation. This means that gene will be abnormal in all the cells of the offspring's body. Eventually, the offspring will develop a second mutation in one of their cells, and this will lead to cancer. In addition, because that offspring has that mutation in all cells of their body, it will also be present in their germ cells, and therefore they can pass it on to the next family member, and therefore this tends to be a family which has lots of colorectal cancer, all because of this germline mutation. Another example that also involves the GI tract is familial adenomatous polyposis, or FAP. This is a germline mutation in the APC gene, another tumor suppressor gene. It always, 100% of the time, progresses to colon cancer. In fact, the risk of colon cancer is so high that people born with this germline mutation have a colon removal or a colectomy surgery prophylactically to prevent the development of colon cancer. And then one final example is the Li-Fraumeni syndrome. This is a syndrome of multiple malignancies that occur at an early age. It's sometimes called the SBLA cancer syndrome. SBLA stands for sarcoma, breast, leukemia, and adrenal gland. These are all places where tumors can develop. This is caused by another germline mutation, but this time in a tumor suppressor gene called TP53. This codes for tumor protein p53, which delays cell cycle progression to allow time for DNA repair. So once again, people inherit one copy of the TP53 gene that is abnormal. Over the course of their lives, in some cells in their body, the other copy becomes abnormal, and that leads to cancer. Now let's talk about mosaicism. This is a genetic term that refers to gene differences in cells of the same individual. So let's imagine that we have a whole bunch of blue cells which are replicating to create daughter cells, and one of them develops a mutation during the course of mitosis. This will lead to a new population of cells which has different genes compared to the other cells in that same individual. Therefore, this individual has a mixture or a mosaic of different genetic makeup, and therefore this is said to be an example of mosaicism. Mosaicism can involve germline or somatic cells. Germline mosaicism can be passed on to offspring. Let's imagine that we have a parent who has three different germline cells that are producing gametes to form offspring, and let's imagine that one of those germline cells develops a mutation. This is an example of germline mosaicism, and any gametes formed from this mutated cell will have a mutation that can be passed on to offspring. Pure germline mosaicism is very difficult to detect. It's not present in blood or tissue samples that are often used for genetic analysis. What this means is that offspring can develop disease that may appear to be a sporadic mutation, but in fact it's due to a germline mosaicism in the parent. So this can sometimes present in families as recurrent sporadic cases of disease in the offspring. What's really happening is that a parent has germline mosaicism that they are passing on to the offspring. In contrast, somatic mosaicism refers to gene differences in tissues or organs of an individual that do not involve the germline cells. For example, some cases of Turner syndrome involve mosaicism. Women with this version of Turner syndrome have some cells that are 45X and are missing an X chromosome, where other cells are normal and have 2 copies of the X chromosome. These tend to be milder variations of Turner syndrome. In addition, there are rare forms of Down syndrome that involve mosaicism. In babies born with these variants of Down syndrome, some cells have trisomy 21, which leads to Down syndrome, but other cells have 2 copies of chromosome 21, which is normal. There is a rare genetic disorder called McCune-Albright syndrome that is often used to illustrate the principle of mosaicism. This is a genetic disorder that affects many different systems. In particular, it affects many endocrine organs. One of its characteristic features is precocious puberty that can start at a very early age. For example, females may menstruate as early as 2 years old if they have McCune-Albright syndrome. In addition, there is fibrous growth that occurs in the bones. This leads to fractures and bony deformity. And then finally, there are characteristic skin findings. The skin becomes pigmented. These children can develop café au lait spots, and the skin pigmentation often has irregular borders that is classically described as appearing to look like the coast of Maine. Shown on the right side of the screen are 2 children with skin lesions from McCune-Albright syndrome. You can see how this child on the left has skin pigmentation with very irregular borders. And in case you don't know what the coast of Maine looks like, this is a map picture of Maine, and you can see how the coast is very irregular, just like this child's skin lesion on her chest. McCune-Albright syndrome is caused by a sporadic mutation that occurs during development of the child. It is not an inherited genetic abnormality. The mutation that occurs is a somatic mutation of the GNAS gene. This codes for the alpha subunit of a G3 protein that activates adenyl cyclase, and when this gene is abnormal, it leads to continued stimulation of cyclic AMP signaling more than is normal. The most important thing for you to remember about this very rare disorder for the Step 1 exam is what's shown on this slide. McCune-Albright syndrome is caused by a post-zygotic genetic mutation. It occurs after fertilization, and therefore only some tissues and organs are affected. In other words, it is said to display mosaicism, and the clinical phenotype will vary depending on which tissues are affected. So to understand this, just imagine that you have a developing fetus and it's got a number of different cells here, and one of them develops this mutation. All the daughter cells of this mutated cell will have the McCune-Albright syndrome genetic mutation, but the daughter cells of these other cells will not have the mutation, and therefore the child will develop as a mosaic of genetically modified cells, some being normal, some having the mutation. If there happens to be a germline mutation that leads to the McCune-Albright genetic mutation in all of the cells, that is a lethal condition. In that case, the entire body is affected. So in other words, cells with this mutation can only survive if they are mixed with normal cells in the individual. What this means is if you see a child in clinical practice with McCune-Albright syndrome, that child must have mosaicism. If the child did not, they would have died and they would not have survived to come in for clinical evaluation. I'll finish this video with a couple more terms that you should be familiar with. The first one is genetic heterogeneity. This refers to when the same phenotype develops from different genes or mutations. For example, different mutations of the same allele can lead to the same disease. In other words, there is not a single mutation that leads to the disease. Multiple different types of mutations can cause the disease. We'll talk more about that in a minute. A second example is when different genetic mutations at different loci on the chromosomes cause the same disease. This also is an example of genetic heterogeneity. So the point here is that for many genetic diseases, multiple gene mutations often cause the same disease. Or another way to look at it is that many clinical diseases have multiple genotypes that lead to those disorders, and this is referred to as genetic heterogeneity. One subtype of genetic heterogeneity is allelic heterogeneity. So by way of review, an allele is an alternative form of a gene. So imagine that allele 1 is formed by mutation X of a particular gene, and allele 2 is formed by mutation Y of that same gene. But both X and Y cause the same disease, and X and Y are found at the same chromosomal location or position, therefore they involve mutations of the same gene. This is termed allelic heterogeneity. It means different alleles or different mutations lead to the same disease. And the point here is that many alleles possess multiple mutant forms that all lead to the same clinical presentation or clinical disorder. So I've summarized this at the bottom. You have one disease caused by multiple genes, but all those genes are variants of a gene at the same location, therefore they are allelic variants, and therefore this is termed allelic heterogeneity. A couple examples of allelic heterogeneity are shown on this slide. Beta thalassemia, which I discuss in the hematology section, involves mutations of the beta globin gene, and there's a wide spectrum of disease severity depending on the mutation. People that are said to possess the beta 0 form of the gene have no function. People who possess the beta 1 form have some function, and I talk about this in more detail in the hematology section. Cystic fibrosis is another example of a disease that displays allelic heterogeneity. Cystic fibrosis is caused from mutations of the CFTR gene, and there are over 1,400 different mutations that have been described that can lead to various forms of cystic fibrosis. Another subtype of genetic heterogeneity is locus heterogeneity. This refers to when mutations in different loci on chromosomes cause the same phenotype. So imagine that we have a chromosome, say it's chromosome number 1 here, and a mutation at this gene right here causes a disease, and then over here we have chromosome number 3 and a mutation here causes the same disease. This is the same disease coming from genetic mutations in 2 different loci on chromosomes, and this is an example of locus heterogeneity. And one example of this in clinical practice is retinitis pigmentosa. This is a rare disorder that causes visual impairment, and there are many different forms of this disorder. There's an autosomal dominant form, a recessive form, an X-linked form. It turns out that mutations at 43 different loci on chromosomes can all lead to this disease. Therefore, retinitis pigmentosa is said to display locus heterogeneity. And I've summarized the key points at the bottom. In locus heterogeneity, we have one disease caused by multiple genes at multiple different locations on chromosomes. And that concludes our video on genetic principles.